Hi everyone. So here we are today with Dr. Dervis Salih, a senior research scientist at University College London. We are in London right now, and we are going to be talking about Alzheimer's disease and its characteristics and the possible treatments about the Alzheimer's disease. So if you wanna make a general introduction to Alzheimer's disease.
Yeah, so thank you very much for the kind introduction. I'm Dervis Salih. I work at the Dementia Research Institute at University College London. This is a new institute. Its purpose is to generate a creative program that can tackle different aspects of dementia. There are 6 institutes throughout the UK which are working together as one institute that's going to have an innovative program to tackle all of the different aspects of dementia. So I work on dementia without trying to be over dramatic. Dementia is a big problem. It's probably the medical challenge of our times. Just in the UK alone, there's around 850,000 people that have various forms of dementia. Alzheimer's is the most common type, and it's characterized predominantly by loss of memory and other executive functions. Throughout the world, there's probably around 40 million sufferers of dementia right now. So just those numbers in itself demonstrates what a big problem dementia is to the world. Over the last few years, the medical research has taken us to a level where we can combat heart disease and a lot of types of cancer quite effectively, which means that people are living longer, and that gives more opportunity for them to develop age-dependent neurodegeneration like Alzheimer's and other dementias. So that's why here we're concerned with dementia. Alzheimer's is the most common form of dementia. It's characterized by 2 main pathologies. That's amyloid plaques, which are protein deposits outside of cells, and intracellular tau tangles, which are inside cells and eventually cause the death of neurons. We don't have any treatments for Alzheimer's right now, and that's because we still don't understand the fine molecular details, and that's probably best exemplified by having an incomplete understanding of what links amyloid and the tau pathology together. Alzheimer's is a disease that develops for decades in a person before it's diagnosed. So initially, through hypotheses such as the amyloid cascade, we believe that the initial start of the disease is with rising amyloid beta, and then that elevation in A beta occurs over years and maybe a decade or 2 before you start to get neurofibrillary tangles, and then that's what starts to cause neurons to die. And then a certain number of neurons have to die before you start to present with memory problems. And that's when a person would go to see their doctor, or family members would bring the person to the doctor saying that there's, something not quite right with his memory or other mental processes. And so to the point of diagnosis, it's already been a few decades where the disease has been developing and killing neurons. And so if 20% of the neurons are lost on diagnosis, then the disease has already progressed quite far. So right now we don't have treatments for Alzheimer's. There are drugs that help to keep the mental capacities quite high by preserving the levels of neurotransmitter to help with daily processes that we need. But we don't have any disease-modifying drugs that actually slow the development of the disease, that actually slow the neuronal death, the loss of the neurons. And so my research is trying to understand the early molecular mechanisms in Alzheimer's, particularly relating to microglial cells. So you might be aware of microglia already. They're the innate immune cells of the brain. And the reason why I'm particularly interested in them is because over the last 10-15 years, genome-wide association studies have shown that a number of DNA variants or DNA polymorphisms are present in genes that are related to microglia. So genome-wide association studies, how they work is that they take large populations of people with Alzheimer's and also control patients that are age matched, gender matched, ethnicity matched, and then they look for DNA variants, polymorphisms, single nucleotide repeats, that are associated with dementia in that particular population. So various studies have highlighted a number of risk genes which have polymorphisms in microglial expressed genes. So this research is just beginning. It's in its infancy. We've identified probably around 40 to 50 risk loci that are mainly in microglia, and somehow they're related to the development of Alzheimer's or altering the risk of developing Alzheimer's. There are a number of groups throughout the world that are studying these genes, such as our labs here and also Christian Haas's group, amongst others throughout the world, that are studying the role of these microglial genes in the actual different cellular phenotypes that the microglia can present. One of the genes that we've been focusing on is TREM2, and knocking down TREM2 alters a variety of different cellular processes in the microglia, such as phagocytosis, the ability of microglia to engulf either damaged agents, damaged neurons, or even synapses. And other things that microglia do is release cytokines that can be pro-inflammatory or anti-inflammatory. So one of the papers that we're working on right now shows that if you have a loss of function of TREM2, then you lose some of the anti-inflammatory and phagocytic processes of microglia, which could have a role in disease.
So I guess the question here I want to ask is the early detection of amyloid beta levels possible in a possible patient that is actually developing this disease, or has there been any study going on?
So there are lots of studies going on that are looking for biomarkers, but also to measure A-beta and other changes that you get in the cerebrospinal fluid of patients. That's actually a key point to try and detect the disease as early as possible. It's quite difficult to do that, and if you imagine that you need to do that on a population level with often procedures that are quite expensive, such as MRI scanners and PET imaging and things like that. So it can't be done routinely, and that's one of the challenges, to identify the earliest changes that can be detected in people without being too invasive as well. Because if you imagine you have to access cerebrospinal fluid, that's not a routine procedure that can be done. It can be done safely, but it's not trivial.
You are working with transgenic mouse models, but we all know that there can be limitations working with them. What are some limitations that you have experienced working with them?
Yeah, so that's correct. We use mouse models because they breed well, we understand their genome really well, and we can make genetic modifications that allow us to study different aspects of disease. Actually, one of the reasons why we don't have good treatments for Alzheimer's right now is because the mouse models, they don't fully recapitulate all of the different stages that you see in human disease. In broader terms, we can engineer mouse models that have either just the amyloid pathology or the tau pathology, but not both of those together. So even within a more simpler organism such as a mouse, we can't see which molecular or cellular processes are connecting the amyloid to the tau pathology. And so, as you would expect, or you might not expect, but the results that we see is that with the amyloid animals, there isn't really significant loss of neurons, but with the animals that show the tau pathology, there's a much more significant loss of neurons. So the way that we can think about it is that the amyloid models representing the earlier stages of the disease, and the tau the later stages of the disease, and somehow we need to understand the part in between, how rising amyloid leads to tau-dependent death of neurons.
So what is the right research path that you think is needed to be followed at this point?
Yeah, so everybody would answer that question differently. There are people that are focusing solely on neurons, different types of neurons, how they're affected by the different pathologies. The genome-wide association studies are pointing towards significant roles in microglia, so the microglial biologists like myself would think about that differently. The hypothesis that we're currently testing is that the response of microglia to the amyloid pathology is actually a major determinant in whether the organism would go on to develop tau tangles or not. So in a healthy individual, the microglia would respond adequately to rising A-beta and to early amyloid plaques, and try and limit the damage to the neurons, maybe by taking away damaged synapses or other damaged parts of neurons. But if there's a problem with the microglia, for example, patients have got DNA polymorphisms that can't deal with the amyloid damage properly, then that elevated amyloid would eventually lead to tau tangles over years, and then you would get more severe stages of Alzheimer's.
Okay.
So with single-cell sequencing, Beth Stevens at Harvard has shown that the microglia are in fact heterogeneous. There's at least 9 different populations of them, and at different stages of development in different regions of the brain, they're able to fulfill different functions. They've also done some very interesting work in identifying changes to microglia with age as well, so that they can see as the organism ages that the gene expression changes within the microglia. And they've also challenged the animals with a demyelinating agent, and that changes the response of the microglia again. So microglia adapt very strongly to the developmental state they're in throughout life, and also we think that they respond differently to different lesions or damages that you might get into the brain. Other groups are working on similar approaches, and we're beginning to understand the genetic pathways within microglia that are associated with disease with amyloid plaques versus perhaps other diseases and normal states and aging. So now the challenge is to understand how those pathways are connected and whether that can help us identify the disease at an earlier stage, or also perhaps identify which levels of these signaling pathways can be targeted for therapeutic advantages. Mm-hmm. So right now, the genome-wide association studies that people have done have pointed to, I said, probably around 40 or 50 different risk loci. In a number of cases, the DNA polymorphism is outside of genes, it's intergenic. So we don't know what that DNA variation is doing to the gene per se, or even to what gene it might be associated with. There's probably a couple of dozen cases where we know that the DNA polymorphism affects a particular gene. Those genes, although we've groups around the world have been working on them for 5 or 10 years maybe, we still don't have good targets to manipulate those genes. So some proteins are more druggable than others, and so if we can understand those pathways better, we might be able to find molecules where we can inhibit that molecule or activate that molecule more effectively to modify dementia. So
bringing it back a little, you talked about how the gene expression in microglia changes as the age progresses, right? Did this study include analysis of like a control group between the transgenic models? Like how does the gene expression in microglia differ from a control group, I would say, in this case?
Yeah, so this particular study, it was done with just control animals.
Oh, okay.
And they did have males and females. Other groups have done it with animals that show amyloid pathology as well. They see that microglia responds quite specifically to amyloid damage, and they coined the phrase, which is disease-associated microglia, actually amyloid-associated microglia. And that seems to engage certain genetic pathways within the microglia. Beth Stevens' group also saw some aspects of that in their single-cell sequencing as well. I think we still need to understand those genetic pathways better. That's one of the reasons why we did our latest assay as well. we found that microglia responds very strongly to amyloid pathology, and that there's a specific genetic network of microglia, and we're trying to understand how that network is connected together, and part of it might be driven by this transcription factor SPI1. Alison Goet's lab recently in America showed that DNA polymorphisms actually modulate the levels of this transcription factor SPI1. Transcription factors are master regulators of DNA, so they control how DNA is expressed into RNA and protein. So just by polymorphisms affecting one transcription factor, it has big knock-on effects for all of the genes for the gene expression program that's controlled by SPI1. So we think that the genetic network that we've identified in our amyloid mouse model is also in part regulated by SPI1 as well. So SPI1 might be connecting together these genes. So now we can use this information to flesh out the pathways further to see if we can find more druggable targets.
I have one last question. So recently, precision medicine has been playing a big role in curing or trying to find treatments, precise treatments for other diseases like cancer. Do you see the future in the research field in Alzheimer's disease using this
so personalized
personalized medicine.
Yeah, I think that's a very good point. We tend to use the term Alzheimer's disease, but this disease, it might develop in different ways in different people, and we're still beginning to understand how that happens. At the genetic level, it's probably that different combinations of polymorphisms alter risk differently in people, and we still need a better grasp on that. So there are, lots of people actively working on that, developing polygenic risk scores, people like Valentina Escott-Price at Cardiff and others. So it's still in its fairly early stages, but I think, that's definitely a good way to go forward as well.